Welcome everyone to Medicine Grand Rounds, and this week's or this presentation of Medicine Grand Rounds will be the Fialkow Scholar Award presentation and lecture. And the Fialkow Scholar Award was established by the Department of Medicine in memory of Philip and Helen Fialkow. And Dr. Fialkow was the chair of the Department of Medicine from 1980 to 1990, and then went on to become, and then transitioned from chair of the Department of Medicine to dean of the School of Medicine. And he was always very interested in supporting the career development of junior faculty. And this award is really the highest award that is given to a junior faculty member at the rank of assistant professor in the Department of Medicine. And he sought to really identify mechanisms to really advocate and support the development of junior faculty members. And I think our selection for this year's Fialkow Award is a prime example of what Dr. Fialkow was trying to support. Unfortunately, both Dr. Fialkow and his wife Helen Fialkow died tragically in an avalanche while trekking in Nepal, and this was established by the Department of Medicine to honor their memories. Each year, the Department of Medicine solicits nominations from division heads for the annual Fialkow Award winner. And you can see a list here of past award winners. And then a committee is convened to vet the nominees and select the winner for that year's Fialkow Award. And our recipient this year is Andrew Stergachis, who's an assistant professor in the Division of Medical Genetics. And to introduce Dr. Stergachis, I'll call upon Dr. Gail Jarvik, the division head of the Division of Medical Genetics. Gail?
Thank you very much, Conrad. It's really my pleasure to introduce a stellar faculty member in the Division of Medical Genetics, and also to take a moment to honor Dr. Fialkow, who was very helpful to me when I was a junior faculty member, actually. I'm very, very pleased to introduce Andrew to you today. Andrew has a long history with us in medical genetics, even though he's only been on our faculty for a couple of years, in that when he was a high school student, he volunteered in George Stam's lab. Then he moved on to the University of Chicago, where he completed a chemistry degree and a biochemistry degree with honors before we were able to recruit him back to our medical scientist training program. And he completed his MD and his PhD in genome sciences as part of that program, where we overlapped, and he did some work in medical genetics at the time. And despite our best efforts to keep him here, he went on and moved to do internal medicine at Brigham and Women's in Boston, completing his internal medicine and also medical genetics in the Harvard Combined Training Program. So he's board certified in both internal medicine and medical genetics. This award is one of many awards that he's won, over a dozen highlights, including, besides this one, the Burroughs Wellcome Career Award, the NIH Director's Early Independence Award, and the Pew Biomedical Scholars Award. He has a wonderful set of publications, over 30 publications, including first or senior authored publications in Science, Nature, Nature Genetics, and Cell. In addition to his research, he does clinical work in medical genetics, where he sees all comers, I think, but also a focus on cardiovascular disease. So he is a true triple threat. I should say also his both internal medicine and research skills have been incredibly helpful to us in the undiagnosed disease program, where we really see both sides, the clinical acumen as well as the research acumen. His title today is Moving into the Unknown, the Next Stage of Genetics and Genomics. Andrew.
Thank you very much for that very, very kind introduction, probably a little bit too kind. So let me just start sharing my screen. So once again, thank you very much for the honor of having this award. This is truly an honor to be named as a Fialkow Scholar, especially as was highlighted, Dr. Fialkow's history as a leader, not just here at the University of Washington, but also in the field of genetics. So I have no disclosures for this presentation. So the goal of this talk today is really to address areas of uncertainty within genetics and genomics, as well as how current technologies are enabling us to address these areas of uncertainty directly head on. So because this is medical grand rounds, I'm going to start off my talk today with a case presentation, which will highlight some of the areas of uncertainty that we face in the genetics clinic today. So this is a patient I saw in the cardiovascular genetics clinic several months ago, and she had originally presented to the emergency department with shortness of breath. Although she was diagnosed with influenza as the cause of her shortness of breath, an ECG was nonetheless performed in the emergency department and demonstrated a Brugada type 1 pattern, which prompted a referral to see myself as well as others in the cardiovascular genetics clinic. Her past medical history was notable for a syncopal episode several years prior to this presentation. And then her family history was notable for several first degree, or her brother who had syncopal episodes, as well as a fourth degree relative with sudden death in the 40s while playing basketball. So just given this presentation alone, you can give her a diagnosis of probable or definitive Brugada syndrome using sort of standard criteria, which are used for scoring different pieces of the clinical as well as the family history. However, the question that really was posed to us is, what is the genetic cause of her Brugada syndrome? And specifically, we want to identify a genetic cause so that we can initiate carrier screening in her relatives to figure out whether or not they were at risk. It's important to recognize that some patients with Brugada syndrome have normal ECGs at baseline. So having this genetic information is really actually the best approach for doing carrier testing in this patient population. So before I go into her specific genetic testing results, I want to discuss how we choose which genes to test for. So confusingly, genetic alterations in over 21 genes have been reported to cause Brugada syndrome. However, many of these disease gene associations have really minimal, if any, evidence for being associated with disease, despite the fact that they're being used clinically to test for Brugada syndrome. So really what we want to do, and what other people have been trying to do, is understand which of these genes are truly associated with different diseases. One of the resources that have been doing this is a resource called the ClinGen Consortium, and they've undertaken a process of actually using evidence-based guidelines for classifying disease gene associations. And this has really markedly improved the standardization of clinical genetics testing around the country, and it's being continuously updated as we learn more about our genome through future research. So for example, a recent effort focused on Brugada syndrome found that of the 21 genes above, only SCN5A had definitive evidence for causing Brugada syndrome. So another piece of background I want to go over before I discuss her genetic test is the nomenclature that we use to actually classify genetic variants. So variants are often split into either common or rare genetic variants based on their prevalence within the population. Notably, the far majority of both common and rare variants do not cause disease. And most of medical genetics I'm going to be talking about so far is really focused on these rare variants down here. And we have actually further nomenclatures that we use to classify rare variants based on their evidence linking them with disease. So actually in 2015, the nomenclature used for classifying these variants was standardized by the American College of Medical Genetics and Genomics. And really this new standardization includes 5 different classes of variants, as well as a scoring rubric that's used to determine which classification each variant should receive. For example, variants can be labeled as pathogenic or likely pathogenic if there is sufficient evidence to say that these variants actually cause disease, or benign or likely benign if there's sufficient evidence or strong evidence to say that this variant does not cause disease. And then there's the variants of uncertain significance, which is sort of the middle class where there's really conflicting or no evidence to determine whether or not a variant causes or does not cause disease. So it's really important to recognize that these VUSs are a major clinical problem. They limit our ability to give accurate genetic counseling regarding patients' risk, and also to enroll patients in trials that may be pertinent for individuals with a genetic determinant of disease. And as more people have undergone genetic testing over the past decade, more rare variants have been identified. And since we know very little about these rare variants, they're often classified as VUSs. However, it's really important to recognize that VUSs are likely, or the majority of these are going to be reclassified as benign or likely benign. It's really just a matter that we just do not have enough information about them to make that classification. However, there are some in here that are putatively pathogenic, and we really need to understand which one of these are actually pathogenic and which ones are benign. And because of this ambiguity, we really do not recommend testing other family members for VUSs to inform the disease risk of their family members. Okay, so now back to this patient. So we sent off genetic testing on her, and we found a VUS in the gene SCN5A. So this is a variant classified, as I mentioned, a variant of uncertain significance. And the reason why this is classified as a variant of uncertain significance really boils down to 3 points. One, this variant is absent from large control database cohorts. However, it's really important to recognize that current population database cohorts do not adequately reflect genetic diversity in individuals of non-European ancestry. And this patient happens to be of African-American ancestry. Furthermore, this variant is classified as VUS because it's never been seen before in someone with Brugada syndrome, so we can't leverage other patients to better understand what's going on here. And third, this is labeled as a VUS because there's no functional evidence regarding how this variant impacts the function of SCN5A. So the next logical question is, how do we resolve this? How do we actually go about resolving which of these VUSs are likely benign or potentially causing disease? Really, there's large efforts right now underway to include more diverse populations in our reference maps of genetic variation, which is going to be essential to addressing this first of the 3 challenges that I listed below. Second, I think really data sharing is essential to ensure that we can recognize when a variant is seen in multiple individuals with disease. And I'll go over a little bit later sort of one of our approaches for addressing this. And finally, it is really paramount that we leverage this emerging high throughput functional assays that are being performed to inform variant interpretations. And this is what I'll go over in the next couple of slides. So really, international efforts right now are underway to perform functional studies on every possible genetic variant in all medically relevant genes. And I think this is a lofty goal that people have set, and I think it's very awesome that this is existing and is potentially going to happen within the next several decades. And the faculty here at the UW have really been leaders in this field, including individuals like Doug Fowler, Leah Starita, and Jay Shinjuri, among others. And given these efforts, we really wanted to determine whether or not we could use these high throughput functional assays to solve the pathogenicity of this particular patient's uncertain variant. So thankfully, Andrew Gleiser, as well as Dan Roden at Vanderbilt, had actually recently established a well-validated high throughput patch clamp assay to identify variants that disrupt SCN5A function. And so specifically what you're seeing here is this is a variant that disrupts function in red versus the wild type, which has a normal function here in black. And these would be variants with some sort of intermediate impact on the function in yellow as well as blue. Andrew then tested the function of this patient's particular variant using this high throughput assay and demonstrated that this variant resulted in complete loss of function, as is shown here in red, which is consistent with other pathogenic variants in this gene. So now using this functional data, we, in addition to other features about this variant, we can reclassify this variant as actually likely pathogenic and use it clinically for cascade testing. However, this is just one patient, and we wanted to know if we could do a similar approach to resolve VUSs for a much larger number of patients that we're seeing in the genetics clinic here at the University of Washington. So to address this, we actually founded the Brotman-Beatty Institute Clinical Variant Database earlier this year, which is a collaboration between the genetics clinics at both the University of Washington as well as Fred Hutch to create a repository of genetic variants encountered clinically at these institutions that would connect patients with rare diseases to researchers doing high throughput functional studies and developing diagnostic and therapies for rare diseases. This work has really been possible thanks to both funding from the BBI, as well as the tireless efforts of both Andrew Folta, as well as Adriana Cedeno-Cortez here. So to date, this database includes over 2,200 participants, which include 2,600 variants, which are EHR linked, meaning that we actually have the health record information as for these variants. And you can see that we're steadily adding patients at a brisk pace to this, or participants at a brisk pace. Using this database, we find that about 50% of the variants that we encounter clinically here at the UW and Fred Hutch are indeed variants of uncertain significance. We've been teaming up with researchers around the world to use these emerging functional data to help determine the pathogenicity of these variants, and would be happy to collaborate with actually anyone watching this who is interested in working with us as well. In addition, we've also been working with ITHS to incorporate this database into LEAF to enable cohort discovery for UW researchers who are hoping to use germline genetic data to identify different cohorts for studies. So going back to the objectives of this lecture, I hope that I've at least imparted upon you in the first part of this talk, how we can recognize some areas of uncertainty in clinical genetics, as well as some of the steps that we, as well as others, are taking to resolve these areas of uncertainty. However, it is important to recognize that everything that I've been talking about so far has largely just been focused on a very small fraction of the human genome, which is the 1% of the genome that encodes for genes. In the next part of my talk, I'm going to switch gears and start talking about how we can understand the other 99% of the genome. However, to do this, is important to understand how the genome is actually packaged inside the nucleus of the cell, as this packaging is actually directly tied to the function of various genomic elements. So first off, I just want to state the genome does not look like this when it's inside of a cell. Rather, the genome is actually packaged into something called chromatin, which is comprised of various proteins that are bound that bind the genome, such as nucleosomes, polymerases, and transcription factors. This architecture of how these proteins are bound is really fundamental to the function of the genome. For example, critical regulatory elements, such as this one here, within the genome are often bound by specialized proteins called transcription factors that really impart the function to these elements. So genetic disruptions to these regulatory elements cause many rare as well as common diseases, and targeted alterations to these elements are currently being used for therapies that really have the potential to cure a lot of rare diseases, including ones such as sickle cell anemia, as well as beta thalassemia, where trials are really in the late stages for doing that. So how do we actually map these regulatory elements? This is pertinent to a lot of what I'll be talking about in terms of my ongoing research right now. So the location of these elements along the genome is often mapped based on their sensitivity to enzymes that non-specifically cut DNA, which is employed in methods such as ATAC-seq as well as DNase-1-seq. These enzymes will cut, and then the short DNA fragments that are released by these are sequenced using short-read sequencers such as standard Illumina sequencing. You can then take these small DNA fragments that are released after doing this and map them to the genome, and what you identify are these regions of hypersensitivity in terms of these are areas which are hypersensitive to these enzymes and ergo are actually regulatory DNA. And this marks promoters, enhancers, insulators, and other sort of regulatory elements along the genome. In work that I did during my PhD with John Stomatoyanopoulos really identified that these regulatory elements occupied around 100,000 to 250,000 sites per cell type. Furthermore, it's important to recognize that these regulatory elements are highly dynamic. If you were to follow a cell during development or differentiation, you would find that on average, around 50% of its regulatory elements that are used by that cell change in really less than 2 weeks, really indicating that this is something which is essential for the patterning that happens during development as well as disease. However, despite sort of, I think, really the benefit that these methodologies have actually had in terms of identifying where these regulatory elements are along the genome, there are some significant limitations which have arisen over the last decade in terms of how we actually map regulatory elements genome-wide. And really, I think one of the best ways of illustrating that is the fact that when we're using scissors to identify where these regulatory elements, we're actually inherently destroying the chromatin fiber that we're trying to study in the process of actually studying it. So as a result, rather than getting sort of this larger view of what's going on and what's bound to that molecule of DNA, we get a very myopic view of only a given spot of the DNA at a given time, and unable to really recognize is how different proteins are bound along that same molecule of DNA simultaneously. Furthermore, because these technologies have really relied on using short-read sequencing, which is inherent to cutting and creating these small short fragments of DNA, they've been unable to map gene regulation along complex genomic regions, which I'll get into a little bit more as to what we're sort of missing in terms of that. So really the question which we propose, which is how do we actually move beyond using scissors to mark where gene regulatory elements are? And can we do something else where rather than cutting at sites of interest, we instead spray paint them? And so essentially what I'm saying here is that you use an enzyme that will preferentially modify DNA that is not bound by a protein. What that does is essentially leaves a stencil of where proteins are bound along an intact DNA molecule that you can then read out. And I'll go over sort of the methodology of how we do this in a couple of slides. So how to accomplish this? So thankfully, there's several decades of literature demonstrating that you can actually use a class of enzymes called DNA methyltransferases. And if you use these, they'll specifically and preferentially methylate these open areas. However, one of the major challenges is that the enzymes that were used to date were using these sequence-specific DNA methyltransferases, which really had poor resolution. Over the last decade, though, in bacteriophage, there's actually been some discussion regarding non-specific DNA adenine methyltransferases that could potentially solve a lot of these problems. And two really major benefits of this class of enzyme are that one, since they're non-specific, they essentially give us resolution at every AT base pair in the human genome. And since AT base pairs make up 59% of the human genome, that's near single nucleotide resolution. The other major benefit is M6A, which is the actual modification on adenine, which is made by these enzymes, is not a native methylation mark that's used in humans. As a result, if we see this methylation on our DNA, we know that we caused it to be there, and it's not necessarily something that was there natively within the cell. So how do we do this method? So essentially what we do is very similar to traditional approaches for mapping chromatin. We isolate nuclei. We then treat it with this enzyme. And then what we do is we just take this chromatin, which has been essentially stenciled with this enzyme in terms of these purple dashes here. We remove the proteins from it at this point, and then sequence the DNA. And I'll go over sort of how we're actually doing the sequencing process. And we're doing this actually with an instrument called the Fialkow hi-fi sequencer. And so just a little bit of background in terms of this sequencer specifically is it ends up giving us sequencing accuracy that's about greater than 99.9%. And our read lengths are only around, say, 16 to 21 kilobase pairs. Importantly, this type of sequencing is PCR-free. So that means that these methylation marks that were imprinted upon this DNA molecule are not lost through subsequent PCR amplification steps. Furthermore, one of the benefits of the newer classes of these single molecule sequencers, such as nanopore sequencing as well as Fialkow sequencing, is that they not only can identify the DNA sequence, but they also have the potential to identify DNA modifications, such as methylation. For example, with the Fialkow sequencer, as the Fialkow polymerase is going through and actually sequencing a read, when it encounters a methylated adenine, it actually causes the polymerase to pause a little bit. And that pause can actually be directly read and interpreted as the methylation being present on that specific adenine base. So this is actually what we're leveraging to get these single molecule chromatin maps. So I'm going to walk you through this figure because this is going to be an important figure in terms of understanding what our data looks like and how we're mapping chromatin. So what I'm looking at right now, this is around a 10 kilobase region of a genomic of the human genome. And what you're seeing here is that each of these gray bars is actually a single piece of DNA. And what I mean by that is that if you were to imagine that we started off this experiment with 100,000 cells, there would be potentially, since we deployed humans, 200,000 molecules of DNA, which could potentially map to this spot of the genome. And this is one of those 200,000 molecules. And along this piece of DNA, what I'm marking here in purple are all the areas where our methyltransferase was able to methylate. And what you can appreciate is you see these footprints or these gray areas between these dense purple, which essentially mark nucleosome footprints or dinucleosome footprints. Also, you can appreciate that you have these larger areas here, which represent accessible regulatory elements, which are these larger patches of purple, as opposed to these sort of smaller intranucleosomal linker regions, which can be appreciated here. So what we're able to do is on a single chromatin fiber or molecule of DNA, we're able to not only map where the accessible sites are, but also able to map where the nucleosomes are bound on that molecule. One thing that you can appreciate in looking at this type of data is just the large degree of heterogeneity that exists. And this is heterogeneity that is biologically relevant because this is what the cell is seeing. For example, on this fiber, this site appears to be closed down and is rather bound by a nucleosome instead. So the other thing you can appreciate is if you actually zoom in on one of these loci, such as this one right here, we zoom on the site, is not only are we able to see nucleosome footprints, but actually we can see individual transcription factors which are bound on a per molecule basis. For example, this is a known binding element for the transcription factor CTCF. And you can appreciate that on these top 10 or so reads, it looks like CTCF is giving a footprint, versus on these bottom ones, it's not. So it really enables you to get this single molecule understanding of what's actually going on in terms of gene regulation at a given locus. Another benefit of sort of the fact that we're using long read sequencing is that we actually can haplotype phase our data. And I'll demonstrate sort of how this is useful when looking at, say, the X chromosome. So this is, bulk data. If you're looking at this particular promoter on the X chromosome, this is the exist gene. This is a gene that's actually selectively expressed from the inactive copy of the X chromosome. And now if we look at our fiber-seq reads, but actually bin them into those that are from the inactive versus the active X chromosome, we can appreciate that this promoter for the exist gene is actually being selectively activated on the inactive as opposed to the active copy of the X chromosome. In contrast, this upstream CTCF binding element appears to be actually bound on both of them, although a little bit better on the inactive as opposed to the active X chromosome. Overall, using this approach, we can actually phase around 80% of our fibers, really giving us the resolution to look at the genome at a much better detail, especially in diploid organisms such as humans. So really what I'm sort of trying to argue here is that, by moving away from using scissors to sort of map gene regulation to using spray paint, we can essentially get a much fuller understanding of how different proteins are bound along that same molecule of DNA. So with this, what I really wanted to address first is, what are we missing in terms of our understanding of gene regulation that we can pick up now using this different technology? And so just a little background in terms of this, one of the areas of the genome for which we actually know very little about in terms of gene regulation is actually segmental duplications, which comprise around 7% of the human genome. This is actually some of the fastest evolving areas of the human genome that underlie many human diseases as well as human-specific traits. However, due to the actual repetitive nature of these elements, they've been really challenging to map to uniquely using short-read-based approaches. For example, this is a genomic locus, which includes the Notch2NL gene, which is involved in multiple different human diseases. And as you can appreciate, if you just map short-read data to this area, you see this large empty area. And really the question that comes out of this is this area truly contain no chromatin accessibility? Or is there actually something here that we just cannot study using traditional short-read approaches? So to address this question, we really wanted to ask, how can we actually more quantitatively assess chromatin accessibility using our single molecule data? And really this falls down to a simple question, which is how do I say on a single molecule data that this larger patch of accessibility is actually an accessible regulatory element as opposed to an intranucleosomal linker region? And to address this question, Mitchell Volger, a postdoc in my lab, leveraged a semi-supervised machine learning approach to actually characterize our single molecule data in each patch of accessibility there as to whether or not it was an intranucleosomal linker region or an actual accessible patch of chromatin. So this is a very zoomed out view where essentially what he's doing here is coloring each patch as to whether or not they're in lavender as an intranucleosomal linker region or in red as opposed to an actual accessible patch of chromatin. And this is just using our single molecule data at this point when we're actually characterizing this. If you then aggregate all these sort of red dots or red lines together, what you get is a profile that looks like this. Essentially, these are sort of peaks where it looks like on our single molecule data, there's actual accessibility at that site along the genome. We can then compare this to standard bulk methods of chromatin accessibility or CTCF occupancy, which is a common factor that's found at many of these sites. And what you can appreciate is that many of these sites match up perfectly in terms of both the bulk data as well as our single molecule reconstruction of the bulk data. However, there are some notable exceptions. For example, this peak right here that we're identifying was not identified in any of the bulk data. And when we actually look into what's going on here, we can see that this actually overlaps a retrotransposon, which is a notoriously challenging element to actually map to using short-read data. So now going back to this locus I showed you earlier, we can ask, well, now if we look at our fiber-seq data at this locus, are we able to actually identify any regulatory elements present within the segmental duplication? Doing this, what you can appreciate down here is that the answer is yes. We can actually identify several novel regulatory elements that are being present here within this Notch2NL locus. And really what this now gives us is a map of how to study gene regulation in some of these more challenging and some of the most sort of freshly evolved areas within the human genome. When we do this genome-wide, what we can identify is that actually over 10% of all the accessible regulatory elements that we identify using fiber-seq have actually never been seen before, really indicating, and this is just in one sample, indicating that there's a large amount of sort of discovery yet to be had in terms of looking at gene regulation in some of these more challenging portions of the human genome. And these are really, as I mentioned before, some of the sort of fastest evolving areas as well. So everything I've talked about so far is sort of using the current build of our genome, which is HG38. But really what we wanted to ask the question is, can we leverage some of these newer genome builds to really get a complete understanding of chromatin along an entire human genome? And so to this, we're really turning to really seminal work done by Evan Eichler, Adam Philippi, and Karen Miga over the last couple of years in terms of actually the first map of a complete human telomere to telomere genome. And it's important to recognize for this that this map added 220 megabases of new DNA sequence, which is equivalent to the size of chromosome 2. So a lot of new DNA was actually uncovered with this, which is a large amount of repetitive areas, such as the centromeres, segmental duplications, as well as the ribosomal DNA and the acrocentric chromosomes. So what we wanted to do and what we did was we applied our fiber-seq technology to CHM13 cells, which is the same cell line used for the telomere-to-telomere genome assembly, and simply asked, can we actually now map chromatin to these areas? And the answer was yes. So we were able to, and I'll go over in the next two slides, show that we can actually map chromatin along all the telomeres and centromeres of CHM13. It will give us an ability to look into and peer into really these previously hidden portions of the genome. So this is an example of looking at actually telomere chromatin. So what you're looking at here, this is the 12p telomere, and these are individual chromatin fibers arising from that telomere. This is the actual TTAGG repeat, or telomere repeat array over here. And what you can appreciate is where nucleosomes patterning ends. There's a huge accessible regulatory element here, which actually is very interesting, which I'd love to go into more detail if people have questions. And then it actually transitions into this encased telomere cap. So you're able to actually visualize on a per molecule basis where sort of traditional chromatin ends and this telomere cap architecture begins. Furthermore, which I'm not going to be able to have time to go into, but please take a look at our preprint online, is that we can show is that when we see these, accessible sites along the telomere cap, these actually are preferentially being localized to non-TTAGG repeat structures, which punctuate these telomere repeat arrays, really highlighting a potential role for these non-TTAGG repeats in breaking the telomere cap structure that's essential for encasing the end of our chromosomes. Furthermore, as I mentioned, not only can we look at the ends of the chromosomes, we can actually look in the middle of it now. So what I'm showing you here, this is the actual, very zoomed out. So we're looking at around 10 megabases of genome on chromosome 5. And this is the centromere, which is right here in the middle of this diagram. And I'm showing you on top CpG methylation, where you can appreciate that there's the most of the centromere is hypermethylated in terms of CpG content. And then right in the middle of the centromere, there's actually this dip in CpG methylation, which was noted by the T2T consortium and is thought to be corresponding to the kinetochore binding region. Now, one of the more striking features when actually looking at chromatin across the entire centromere is that most of the centromere is really tightly closed down. You see very little chromatin accessibility throughout it, which is consistent with what you would expect heterochromatin areas to have. However, right in the middle of the centromere, right where this kinetic core is binding, this actually is a highly accessible chromatin, to the extent where this is actually the most accessible chromatin in the entire human genome, which is present right here. However, the type of chromatin here is very distinct from that which you see in the eukaryotic genome, because rather than being sort of this well-organized chromatin accessibility, you actually see this sort of hodgepodge or accessibility, which differs between individual molecules that are mapping to this area, really demonstrating that there's a novel form of chromatin compaction, which is unique to where this kinetic core is binding in the genome. So that's just some high-level views of some of the stuff we can do, really just demonstrating in terms of how we're moving into the unknown, that we really have the tools now to explore these previously unmapped regions of the human genome. However, going back to one of my first points, which I said is, population diversity, exists, and it's important to recognize that. And moreover, some of the work that Evan Eichler and others have done have really demonstrated that population diversity is enriched at these loci, which are some of the more repetitive. So if we want to study, these in other samples, how do we actually study them appropriately? And to this, it really sort of comes down to this question of how can we actually leverage, or can we leverage our data to actually build a genome directly from scratch from each patient or participant or sample that we're doing this fiber-seq on. And this is just highlighting the fact that the CHM13, or this complete telomere to telomere genome, really only represents one individual. So this is actually work by Mitchell and Elliott, and as well as Jane in the lab, where what we did is we took fiber-seq data from a patient with the 46XX karyotype and simply asked the question is, can we take this data as our primary backbone and actually build out their genome directly from scratch using this data? To accomplish this, we supplemented this data with ultra-long-read nanopore sequencing from the same sample, as well as short-read sequencing on the parents of this individual, which I'm just sort of diagramming here, the type of sequencing we had for this. And we leveraged a recent diploid assembler called Fialkow, which is produced out of Adam Philippi's group in collaboration with Evan Eichler's group, the two people, the three who were involved in the T2T consortium. The interesting thing about this is that just using our fiber-seq data as a backbone with these two additional supplemental sources of data, we were able to actually build telomere to telomere assemblies for over half of the chromosomes out of the box. And I think that this is really a testament to sort of how wonderful sort of the Fialkow diploid assembler and these other tools which are being produced out there are, is that you can out of the box get these great assemblies, and really sort of highlighting the hope of being able to make these personal genome assemblies from individuals to being able to better study these more complex regions of the genome in an individual. So now I think what I've highlighted is we have not only the tools to explore, but also to actually map these previously unmappable regions of the human genome. And so with that, I'm going to sort of take the last 5 or 10 minutes of this to really go over one of our approaches of how we can actually apply this technology for better understanding how genetic variants actually impact overlying chromatin. So this actually goes back to an earlier observation that we had, or one of the earlier observations we had when looking over our fiber-seq data. And this actually observation was that if we looked at the HTT promoter, which HTT, for those who don't recognize this gene, this is the gene that's involved in Huntington's disease, is that the actual CAG repeat in this gene overlies on many fibers an accessible element that appears to be this sort of accessibility that is directly overlapping this CAG repeat. And really what we want to ask is, how is chromatin organized, not necessarily around the CAG repeat, but actually within the CAG repeat itself? Like, how are nucleosomes, what's going on inside of that repeat in terms of chromatin organization? And this is not just sort of an esoteric question, but actually has some meaning, because CAG repeats are known to form these slipped DNA structures. And these slipped DNA structures are really involved in the pathogenesis of Huntington's disease by enabling repeat expansions both on the germline as well as on the somatic side of things. And then another important piece of information out there from several decades ago is that longer CAG repeats are more likely to form these slip DNA structures, or sDNA as is being described here. But going back to sort of one of the initial points which I said, which is DNA doesn't just exist inside of the cell as naked DNA. It has nucleosomes and chromatin all bound to it. You can sort of imagine that, well, if the CAG repeat is being bound by a nucleosome, how would it actually be forming this slip DNA structure? So really the question becomes is there something about the chromatin environment here which is sort of causing the CAG repeat being preferentially formed into this non-nucleosome bound structure that may then make it permissive for forming these slip DNA and subsequently undergoing expansions, you name it. So to approach this, actually, so this is work by Stephanie as well as Ben in the lab. They developed the targeted fiber-seq approach to actually not just enable us to look at this HTT area, but actually enable us to look at this across multiple samples in a multiplex manner, really enabling a much higher throughput of this approach than the traditional genome-wide methodology that we use to do this. They then applied this targeted method to a family with Huntington's disease, and specifically this is a father who was affected by it, unaffected mother, and then two children, one who has at risk, so they had the expansion, and then the other one who had a juvenile onset. As the readout in terms of our data is single molecule, we can then just look at what are the copies of the repeat length on a per molecule basis. For example, in this individual with adult onset Huntington's, you can appreciate that the two alleles here, one which is the unexpanded, and then the other which is the actual expanded, versus this unaffected individual, you can appreciate the two alleles which are both unexpanded. Furthermore, you can see that in this individual with juvenile onset HD, you can see that their expanded allele is more expanded than that in the father. And furthermore, there appears to be some somatic variability within the expansion in this sample. Looking now at this sample, what you can appreciate if you look at the chromatin architecture on a per molecule basis, that actually this repeat expansion is notably contains large areas of accessibility that are filling up at least half of the actual repeat in and of itself, with the other half being bound by a nucleosome as appreciated in these areas. We can then quantify this, and what you're seeing here is that if you have fibers which are unexpanded, so you typically only see an area of accessibility that's on average around, say, 50-ish base pairs or less. However, if you actually expand out your allele, what happens is you now appreciate there's a larger area of accessibility which is filling that allele. And going back to this diagram I showed you here, is what you're seeing here is that actually when these expanded alleles, how chromatin is actually positioned within them is it's actually creating a permissive environment for actually this sDNA or slip DNA formation. So now going back a couple of slides in terms of thinking about, just 2 points I just want to highlight is we've done this again on primary brain samples from patients with Huntington's disease, and we've seen a similar type feature. And then another actually really interesting thing that we've seen is that when we look at other CAG repeats, so not the Huntington's one, but other ones that are present in the genome which are not pathogenic, we don't see this feature there. So it seems to be something unique with how the CAG repeat is positioned within the Huntington's promoter that's causing this. And so now going back a couple of slides, you can sort of taking this into a larger context, what you have is a situation where the promoter is being positioned right here. You have this +1 nucleosome, which is sort of immediately adjacent to promoter, and this CAG repeat happens to be located right in this internucleosomal linker region, just immediately adjacent to this +1 nucleosome. And that positioning relative to the promoter is actually creating likely this environment, which is making it permissive to forming this large accessible element immediately sort of in the CAG repeat, which is really unique to the HTT gene. So just to sort of summarize some of the stuff I've talked about so far, and this is really thinking about where we're going next in terms of genetics and genomics. I think it's really important to recognize that functional data sharing, and really diverse populations are really essential for us to sort of solve this first problem, which I was discussing, which is this variance of uncertain significance. And I really, hope I'd sort of emphasize is that some of the recent advances in genetic sequencing have really enabled us to not just look at, the exome, but really move out into not just the genome, but actually complete genomes and epigenomes. And really, these tools not only enable us to visualize, complete genomes in terms of its DNA, but also how this DNA is positioned within the nucleus in terms of proteins that are bound on top of it, which are essential for the functional output of this DNA. And something which I haven't really gone over at all today, but I really sort of think, is important to recognize is that how do we then take this improved understanding and actually move this into improved therapies? And, this is something where, current therapeutic pipeline is really being developed for many of these diseases in terms of knocking out or adjusting the gene regulation of different disease genes. So having an improved understanding of how this is going on is really going to be essential. So with that, I really, want to thank many, many people for this. Gail already mentioned some of the funding support for this, which has been absolutely essential. I think I've already highlighted everyone in the lab. I think the only people I haven't mentioned so far would be Itse as well in the lab, and then also the former lab members who've been essential for this as well, and then many of our collaborators who've been essential for the different projects as is indicated here. I also want to, highlight not only just the people who are currently working with me on these projects, but also some of the people who've just been instrumental along the way in terms of getting to where I am today. Really, if it wasn't for the wonderful mentorship, of many of the people listed here, as well as many others, I would definitely not be sitting here talking to you today. So I really am, truly grateful for all the time that they've dedicated to me as well. So with that, I would love to take any questions. Looks like there's some.
Yeah, no, thank you, Andrew. I wanted to also add that Andrew gets more than his name just on a plaque. He also gets $10,000 deposited into a fund for scholarly activities. And so a $10,000 award also comes with this Fialkow Scholar Award. And Andrew, I'm just amazed, how the technology has evolved in the last 10 or 12 years, to start really dissecting chromatin structure and these uncoding or the non-coding regions of DNA. It's just fascinating to me as an outsider. But I wanted to ask you, I'll start off with just one question, and there's a few more. Please put your questions in the chat. Is right now, you're focused on trying to dissect out the genomics of, or genetics and genomics of rare diseases. How about common diseases? How do you see this playing out for the common diseases? Atherosclerosis, diabetes, et cetera. How about the biggies and just not the exceptional rare ones?
Yeah, I think that's a great question. That's actually something which we're trying to approach right now a little bit as well through some collaborations. And specifically, we're trying to build up a collaboration to do this for Alzheimer's disease. And the goal of this is, one of the major findings, work that was done, with John Sam as well as Matt Morano, back over a decade ago now, demonstrated that a lot of the GWAS variants that have been identified actually localized to non-coding regulatory elements. And this has really been instrumental in terms of our understanding of how these common disease associations exist. However, one of the challenges over the years has actually been identifying how and which of these elements are actually playing a role. And I think one of the major challenges has been that we've been reliant upon genomes that actually are probably not reflective of these haplotypes. And also, we are only able to look at a very myopic picture of how these GWAS-associated variants are actually impacting gene regulation. So I think that thinking about that from can you better approach this problem by building haplotype resolved maps of gene regulation in diseased tissues is one that we've been really trying to take head on. So I do think that that's useful and good. I think that the challenge has always been is, with anything that's common disease related, is the numbers needed for actually doing the testing is usually a lot bigger. But I think that we're at the level where we can actually start to approach that.
Thanks, Al. The first question that comes up is a question from Carol Baumsteck. It's a technical question, one that I certainly couldn't answer. It says, Andrew, why don't you use M6A MeDip-seq?
Yeah, that's a great question. So we actually did do that. So I'd be happy to. So actually, in our initial paper from 2020, we did exactly that. The challenge with the M6A MeDip-seq, though, is that it's still a short read based approach. So you can definitely do it. And actually, we demonstrated this. We used 5 different non-specific methyltransferases, treated cells with these, and did this. It's an antibody based approach where you're pulling out where the parts of the genome are actually methylated. It will demonstrate that the methyltransferase is selectively going towards sites of chromatin accessibility, but you're nonetheless, since it's a short read based approach, you can't sort out how these different sites of accessibility are happening on the same molecule of DNA. And you similarly cannot map these reads to these complex genomic regions. I think one of the other papers I think was highlighted there is this is sort of an area which is a fun area of research, which is M6A even real in humans? So there's a huge controversy regarding endogenous M6A in humans. It's been argued that it could be possibly present at around on the parts per million, which means that for every million adenines you see, there's around maybe 10 or so of those could be M6A methylated. There's a recent study which sort of has argued that a lot of this is actually contamination from bacteria in a lot of samples. But I think it's sort of an active area of research that's been going on by many different groups. So I probably don't want to weigh too much into that other than saying that, yes, RNA is definitely methylated, but the DNA is less, that area is definitely more complex. So, and it looks like Fuki has a talk.
Yeah.
Yeah.
Fuki says, great talk. Can you expand on this work, how this work could lead to the potential therapeutic or talk about the potential therapeutic implications for Huntington's disease?
That's a great question, one that I've been thinking about in terms of trying to better understand it. I mean, really, I've been viewing this as more, how can we understand how this area is so prone to undergoing these somatic expansions, how to translate that into therapy. I would love to get other people's thoughts, people who may be a little bit more subject matter experts in the Huntington's field. If you're listening to this, please contact me and I'd be happy to talk to you more about that. So I think it's sort of, if you can better understand how it's undergoing somatic expansion, through this process, can you now target this to potentially prevent that somatic expansion, which is hypothesized that somatic expansion of the repeat plays a role in the actual pathogenesis itself. So I think it sort of goes after this question of how do you actually prevent these areas from even forming this accessible chromatin element, which is then being permitted to form the sDNA, which then undergoes somatic expansion, which then potentially is the disease, if that's truly the pathway of the Huntington's sort of pathogenesis.
And maybe just expanding on that a little bit outside just of Huntington's disease, what do you see as the avenues or for therapeutic approaches? What tools would you have to treat these genetic diseases? How do you see it playing out?
I mean, I think, one great example is the recent clinical trials that have been done for, sickle cell disease and beta thalassemia, when you're essentially taking a regulatory element which controls the transcription factor, which is essential for, for actually regulating the gamma globin promoter, and you're knocking out that regulatory element, and you're subsequently sort of helping express the gene. So you're sort of in more simple terms, you're toggling the regulation of a gene to get the phenotype you want. Also work by Andre Lieber and Cheng Li, who I think you guys heard from a couple of weeks ago in terms of his recent grand rounds. Similarly, the approach there is target gene editing to the non-coding genome to a regulatory element to actually correct a disease. And so I think it's really essential for us to have a better understanding of how these gene regulatory elements are situated and how this is playing out for us to even be able to appropriately design these targets. Actually, the gamma globin promoter, which Dr. Lieber's group as well as Cheng Li's group is going after, is a great example of this, because the gamma globin gene is actually duplicated. So you have 2 identical copies, and short read data does not like to map appropriately to each one of those copies. And so to actually better understand, or to understand how each of the gamma globin genes is regulated, you really need an approach like this to really address that question.
Okay, well, we're getting near the top of the hour. So Andrew, the Department of Medicine is proud of your accomplishments, and we look forward to future contributions such as these in the future. We're very proud to have you as a faculty member, and congratulations on the Fialkow Award for 2022.
Well, thank you very much. This is, like I said earlier, this is such an honor. So I really appreciate it.
Okay, thank you.